Hi, this is Gregg Schumer, but Susie, go ahead and get in. You can please discuss the molecular methods work group.
Okay, sounds great. Thanks, Gregg. So thank you to both Eric and Gregg for hosting this webinar. I figured that this would be a great captive audience to just put a quick plug in for this new work group that we've just started up. That falls under the California Water Quality Monitoring Council, and we're calling it the California Molecular Methods Work Group. And this work group really grew out of the recognition that there are a lot of different folks across California who are either interested in or already using DNA or sampling DNA out in the wild, and that there are many of these different groups that are sampling using potentially different protocols and analytical techniques, which means that all of their excellent data that they're generating might not necessarily be comparable. And so additionally, there was an understanding that a lot of different sampling teams are out working across California that might be sampling in similar locations or even targeting some of the same species, and they just don't even know who else is working in that same area. So we really put our heads together to try and come up with a work group that could just help better coordinate all these different activities across California. And so the mission of this new molecular methods work group, of which Gregg and a few other folks from Genidaqs are going to be key players, the mission of the work group is to serve as a clearinghouse for technical and programmatic guidance on the use of molecular methods for bioassessment and environmental monitoring programs in California. So what does that mean? Well, we have a couple of key goals for the work group as it's starting up now and for the next couple of years. And that'll be really to focus on developing some recommended protocols and best practices for DNA sampling and analyses. Also to provide some reference and training materials to the user community. So that's both for experts who are used to using these techniques, but also people who are interested in jumping in and starting out new using DNA-based approaches for bioassessment and biomonitoring. And then, like I said, one of the key goals of the work group will also be to help coordinate research programs across the state to improve method consistency, but also hopefully find new opportunities for collaborating. And so with that, I'll say that right now membership for this work group is open. We already have a great crew of about 40 people that come from a diversity of different backgrounds, industry, academics, some agency, water board, nonprofits. And we're really interested in having a diverse group with a lot of different technical and non-technical background, folks that have been involved in the implementation of novel technologies before for statewide monitoring programs. It would be great to have input on a range of different backgrounds. So if this sounds like something that you'd be interested in, or even if you just want to learn more about this work group, I just encourage you to contact me. My email is here, and I'd be happy to share some more details with you. So that's it from me, but thanks again to Eric and Gregg for giving me a chance to put a little plug in.
Oh, thank you, Susie.
Yeah.
Thanks, Susie. That was really good. I want to join again.
Nice. You're welcome.
Yes, it's nice to see.
Okay, so in the interest of time, I'm going to go rather quickly and attempt to hit the highlights of this, just so we can keep everybody in, and we'll hold off questions till the end, hopefully. So we're going to talk about DNA detection of cryptic, rare, protected aquatic species. We've been working on a fairly large list of species all over the state of California for about 8 years. eDNA comes from biological material that's shed into the environment. If it's an animal, aquatic, terrestrial, and it's from this planet, it should have DNA, and it most likely is shedding some biological material into that environment. If it's in the aquatic environment, it's mixed in with the water and everything else that's in that water, and it's a mixed signal of stuff floating around. If it's terrestrial, like in that bottom right corner there, it's usually a discrete sample of footprint, feces, hair, tissue. It can be actually identified to the individual, which is harder to do in the water, if not impossible. Next slide, please. eDNA, hopefully you're looking at a diagram of a fish in some water with flow from left to right. If there's a small number of individuals and that individual is shedding tissue or material into the environment, there will be DNA within those cells and those tissues. If the water's flowing, those particles will move downstream and they'll settle out like any other particle. It's like firing a bullet. Over a distance, the bullet will just fall out and fall down and hit the ground. Well, the particles of DNA or tissues or whatever's floating around there act much the same way. They will go with the flow, and they will drop out of that system according to physics and dynamics and whatever else is eating it, potentially. Go ahead and click, Eric. You should see the next slide, which is a bunch of fish and a lot more stuff in the water. So the probability of encountering these molecules goes up as there's more molecules to encounter. More biomass equates to more stuff in the environment, and your ability to detect that stuff in the eDNA signal goes up if you're filtering within a distance and you can get a decent volume of water, right? The more organisms there are, the more stuff, the less volume at a closer distance you can get to. This is a stagnant. Next slide, please. If your environment is stagnant, it stops, there's no movement, the system will stratify. You will have to change the way that you sample DNA based on the stratification of DNA. It'll fall out of the system and it'll go down to the bottom. It'll even settle into the soil. In the case of vernal pools, if they get dewatered or they dry out, a lot of that material will be left in the bottom of the pool. Again, the soil can actually be sampled. But in the aquatic environment, if it's a stagnant, non-moving environment, you need to sample a little differently. Next slide, please. The application, especially in California, the eDNA applications are vast and extremely diverse. We go from a desert, pure sand dune desert in the southern portion of our state, all the way to a temperate rainforest in the northwest corner, just in all the environments in between, Central Valley, agriculture, riverine, lacustrine, alpine, coastal forest, wetlands, delta, estuary, you name it. And there are organisms and endangered and invasive species in all of them. And we've been working to adapt techniques to all these environments, slowly but surely. Next slide, please. What we have been working on, our whole philosophy has been from the beginning for the last 9 years. We do three things here at Genidaqs. We figure out where organisms are. That's a big thing. That's very hard when you talk about something like a delta smelt. Where are delta smelt? Well, they're in the delta, but where are they? This is not the easiest question to answer. If you can figure that out, you can move on to things like how many are there and how are those individuals doing. We feel that eDNA at this point deals with just number one. It's where are they? We have not gotten adapted eDNA to the point where we can count the number of individuals, nor can we tell if they're healthy and reproducing. Next slide, please. I keep forgetting to change it myself. There's a lot to cover in eDNA sampling and assay production and filtration types, and there's a lot of information to cover. I'm not going to go over all of it today. For the purpose of this discussion, I'm going to go over the probability of detection, qPCR assay validation, some of the MICE guidelines, data interpretation. There's a little bit of information of all the interlying subjects, but mainly it's going to be about probability of detection and qPCR, assay validation, and data interpretation. Next slide, please. For a really good manuscrict on the critical consideration, I think this is a great title. A lot of the things that everybody needs to consider for doing using environmental DNA to detect aquatic species. They're outlined in great detail in Karen Goldberg's paper. There's also a book out that Pierre Taverlet wrote, which is also good. It's great. Just put it on your nightstand and keep you awake at night. It's a good reading material. These outline everything very well. We try to adhere to the best of our ability to much of what is included in this paper here and in the Pierre Taverlet's book, and of course we adapted it to our own system. We have some differences between what Karen Goldberg does and what we do. Next slide, please. Name principally for the discussions of this conversation, anything that we do here at Genidaqs, we use for a sampling apparatus. We use silicone tubing, a peristaltic pump. It opens and closes, it pinches it and opens it, pinch it open, and it creates peristalsis. which pumps water across that tiny little filter up in the top right corner there, which is called a SteriVex filter. It's an encapsulated filter that you push water across the membrane, and you can cap it, label it, bag it, freeze it, and extract DNA right out of the capsule without having to expose the filter to the environment, which I love. That's a bit different than most people do. Most people use a pour-on-top method and then suck the sample down across the filter. Next slide, please. So survey design. One of our major considerations is how do you design a survey? What do we consider? So well, we consider a lot of things. Mainly, how we sample apparatus, the probability of detecting the organism that you're looking for in its habitat. This is a big deal. This dictates how you're going to sample and what your design will look like. You need to consider the habitat, what type of filter you're using, the volume of water you can actually get out of the environment. Sometimes we sample in rice ditches looking for garter snakes, and we get 30 mLs of water. On the opposite end of that spectrum, we also look for invasive brook trout in high alpine lakes in Oregon, and you can get 4 liters of water through that same exact filter. These are two completely different. This is a huge spectrum of volumes here, so we need to incorporate that into our probability of detection of organism. Replication. How many filters do you need to take? How often do you need to take these samples? Given the life history of this animal, can you make a single observation and determine presence or absence? In every organism that we've tested, no is the answer to that one. No single observation is sufficient. Intervals. Where do you take these samples along your system? Is it a small stream you can jump across, or is it the Sacramento River? What do you need to take into consideration? Discharge of your system. The life history of the organism is obviously important. These things hibernate. Maybe you don't want to sample in the winter. Maybe during reproductive periods you have a greater chance. When is that? Where do they do it? That kind of thing. And then, of course, there's the eternal consideration of budget. Can you do what you need to do for an effective survey? Do you have the money to do it? And then expectations. How important is this to detect? Sometimes it's you go out, and we've encountered situations where they kind of want to know, is it there? Is it not there? Or in other instances, there's government agencies sampling for the DNA of a protected species below private property with water running through it. This is a different expectation. The presence of an invasive species, that has different expectations around it. If you detect a zebra quagga mussel, well, this has different implications than if you detected, something non-invasive but just rare. Delta smelt has implications. Each of the contexts have different expectations and desired outcomes, at least. So you have to weigh that into the design. Next slide, please. For us, this is kind of our graphic of what we go through to determine probability of detection. Volume is volume, which whatever you can get out of the system. The SteriVex has a max volume of 2 liters. When you're in really clean systems, you can get more. It doesn't do you much good, or it hasn't shown. We have not shown that getting more volume through a SteriVex does us any good. We've tested 4 pore sizes of disc filters on the pour-on-top method. We've found in our context with our species and our systems that we're doing eDNA, we have found the SteriVex to work better for us. I like it for a lot of reasons. Suffice it to say, it works better for us. Distance from the source, how far? Now, typically what we do is we take a live car with the target organism, if it's possible, or a surrogate. You put it into the system itself in a live cage. You walk away, you go 1 kilometer away, and you approach the live car taking samples at distance intervals, 1 kilometer, 500 meters, 400, 300, 250, 200, and then you determine at what point you begin to detect the signal from a given biomass, whatever it is you happen to put in the live car. If possible, this is an ideal situation. A lot of times the organisms people are looking for are rare. They can't find them anyway. They need eDNA to detect them in the environment, so calculating a probability of detection is not always possible However, we try to do our best to at least use a surrogate in the same system to estimate the probability of detecting. What your biomass is it a live biomass or a dead biomass? That's a big deal. Dead stuff gives off more, it's decaying rapidly and gives off a greater signal. We've used dead fish as a surrogate for the species, the target species, and you can get really far away from a decaying fish and detect DNA. And of course, underlying all of this is the laboratory, the ability to extract DNA off the filter and analyze the extracted DNA using qPCR and now metabarcoding. For the remainder of this conversation, I'm alluding to qPCR. Metabarcoding is a whole other topic of conversation that can be had at another time during another presentation. I'll discuss it a little bit, but not in great detail. Next slide, please. Okay, hopefully everybody's seeing the determining pod of delta smelt at the CVP. This is the Central Valley Project. I'm not going to go into a great description of what that is exactly. So the delta is on the left side of this picture here is the delta moving off and up to the right corner. That would be the Central Valley Project. So one of the two main canals that transport water from Central California down to Southern California. There is a fish collection facility up in the left side here where they sort fish and they salvage fish and they count them and they try to figure out what's going on and what's being impacted. Well, they do efficiency studies there using delta smelt to figure out how efficient they are at salvaging. We took advantage of this situation and worked with the Bureau of Reclamation to determine the probability of detecting delta smelt at the CVP. This is a unidirectional system. It always moves from left to right. It never reverses direction. The water is always going towards the canals. Water never comes out of the canal and goes back into the delta. So we did the live car experiment with a biomass of 10 fish in each live car, or 50 and 50. We went a kilometer away and approached the live cars at these intervals with these two different volumes from 50 to 300 mLs to essentially get at our probability of detecting a known biomass of delta smelt in a unidirectional system. We are now working on the math to do this in a tidally mixed system. It's considerably more difficult, and it's taken us quite a bit of time, but we're getting close. Next slide, please. So what was it? What did we calculate? So after a lot of work, you're going to have to click through this. Eric, it's an animated slide, so keep clicking. Just keep clicking until you get to the volume at 3. So that you should see a bunch of blue stripes at 300 mLs with a 0.45 micron filter at a 200-meter distance with 100 fish in the live car and one filter. Our probability of detecting that fish was 60%. Now just using basic sampling theory, if you double the number of filters, your detection probability goes up to 84. Keep clicking. It should go to 3, which is 94% probability of detection. And if you go up to 5 filters at 300 mLs, 0.45 micron filter, 200 meters away from 100 fish, you have a 99% probability of detection, which is pretty good. So we're doing this with a couple of other species to verify across species types and water years. The year we did this happened to be the wettest year on record in California, and I don't even know, not more. Next slide, please. So our probability of detection, it matches up with much of what you would read in the literature. We didn't discover anything groundbreaking there. We just determined the probability of detecting the species that our target species in the system we happen to be working in. However, this did come into alignment with what the United States EPA has found. I've been talking with Eric Waits for a year now. We've been working together. He's taking a lot of data points from his Chesapeake Bay Mussel project, and they have calculated the number of samples you need to take at 2 different volumes, a low volume on top and a higher volume on the bottom. The number of replicate samples you need to take per site given the drainage area. So drainage area increases from left to right across the x-axis. Probability of detection goes up on the left axis, and the number of samples across the top from 1 to 10 samples. Hopefully you're all seeing this slide. So you can see as your drainage area gets bigger, you need to take more samples. So the 5 sample size is what we have, the 3 sample size in the unidirectional system is what we settled on. We're moving up to 5 samples, but it's all, a lot of this work is all coming together, and it's all, different groups are starting to come to the same realizations. Next slide, please. Assay validation. So this is quantitative PCR. I don't want to go too much in. Next slide, please. I don't want to go too much into molecular biology because it usually bores people a lot. I love it, but that's because I'm a molecular biologist. The guidelines for publication of quantitative real-time PCR experiments and the design of assays and efficiency levels and cross-reactivity and optimization and validation, all this stuff, they've been really well outlined by a bunch of analytical chemists in 2009. So these guidelines have been well established. We really don't feel like we need to reestablish them for eDNA, so we don't. We just follow these guidelines. Anybody can download this paper. It's free. It's open source. There's no restrictions on who can download this at all. And if you're doing anything with qPCR and you have not already read these guidelines, which I'm sure most of you have, they're doing PCR, I would recommend reading that. Highly recommend it. Next, next slide, please. So this is the basic outline of how we go through this is a red-legged frog, California red-legged frog, which luckily for me was one of the easiest ones we ever designed. I just happen to have a tissue sample, a positive control from every RONID along the western United States. So developing an assay for RONA for this one, this red-legged frog, was actually fun, and it worked pretty quickly. The basics are you get a bunch of vouchered specimen of the target organism, hopefully, preferably from across its entire range, so you can incorporate any genetic variation that is within the species across its range. So you're not just looking at 2 individuals from one pond. Hopefully you would get a cross-section of the entire population, and then you extract DNA. You sequence the DNA. In this case, we did the 2 mitochondrial genes. That's important to note. eDNA is the detection of mitochondrial DNA, not nuclear DNA, which is one of the reasons why you can't do population genetics from eDNA yet. So you generate DNA sequences the old-fashioned way, put it into a Sanger sequencer. They send you back a file. You look at these sequences. You line them up. You find conserved regions of the DNA, essentially a DNA barcode of your species. You look at this barcode on the NCBI nucleotide website, which is also free access, and it has the potential to compare your barcode from your species to every known nucleotide sequence that has been published within this database. And it does a logical alignment, a BLAST, where it compares it to everything known to mankind on this that has been submitted. And you can see if your barcode is in fact unique to your species. And you can go through a bioinformatic basically in silico validation, narrowing down the regions of your mitochondrial gene for your target species that are a good candidate for assay design. The assay design algorithms are also commercially available. Some of them are free. Like IDT has a free. A lot of the DNA synthesis people have free algorithms with limited. It limits the amount you can mess with the algorithm. You can buy them, which is what I did. I bought Primer Express from Thermo Fisher Life Tech. Now it's not Applied Biosystems, which I'm sure many of you realize. I don't know how long it's been Thermo, but I just buy it. I like this. I like the design software, and it allows me to do what I want to do, and it usually works well. Once you have a designed assay, you can go through and validate it, you optimize it, you make sure you give it the right chemistry, the right conditions, how many cycles do you need, all of this stuff. Is it specific to your target? Does it cross-react with closely related coexisting species? How sensitive is your assay? Can you actually detect a minute amount, an amount that is representative of an eDNA sample, like 10 to the minus 4 or 10 to the minus 5th nanograms per microliter? That's not a lot of DNA. Does your assay work at that level? Next slide, please. And then you can begin to test it in the field. Test it on the unknowns. With some caution, you need to move out into the field once you've validated and characterized your assay. Next slide, please. So this is an example of a brook trout assay that we designed. This is just a typical, on the bottom right is a standard curve. You dilute your DNA in a serial fashion. This is a 10-fold serial dilution and allows you to calculate, you plot the slope of this through your points. Hopefully you can pipette. The R squared is a metric of your ability to pipette. The closer you get to 1, the better you are. Onto the plate itself. The y-intercept tells you within how many cycles you can detect the minute amount of DNA. So for this brook trout, you need to run 38 cycles at least. You'll see in the literature some people run their qPCR out to 55 cycles. That's a lot of cycles. I won't talk about that right now. The efficiency of this assay can be calculated as a function of the slope. The slope is 3.49. The efficiency, therefore, is 93.3%, which is pretty good. You can get 100% efficiency on some of these. You get over 100%. You can imagine that. I'll let everybody ponder that one, how you get more than 100% PCR. Okay, next slide, please. The data that you generate off this is inherently presence-absent. The data set is presence-absence. You either detect DNA or you do not detect DNA. There are things that affect your ability to detect DNA, which we tend to take into account during the determining the probability of detection, inhibition. There's volume limitations, there's PCR efficiencies, there are all of these things involved that you need to incorporate into your survey design and in your data interpretation. It's either there, but the DNA is either there or not there, which is a function of CQ. It's a strength of detection. Because this is quantitative PCR, you can actually look at the strength of detection, which is handy when you want to compare the strength of a DNA detection from one place to another. Now, there's a lot of work out there being put towards using a quantity of DNA in your sample to relate it to a known biomass of target organism, which is great if it works out in the long run. We do not do that right now. We simply compare the strength of signals from one place to another, and we do not relate it to an amount of target organism in the system. So this goes back, the data set for us, it relates all the way back to the first question. Where are these organisms? That's really what we're concerned with when we're doing an eDNA survey. Where are these things? Once you figure that out, then we begin to expand the survey and try to figure out when are they there? Which is another big question. Next slide, please. This is another animated slide. This type of data lends itself quite well to occupancy models. You can use the presence-absence data to create an occupancy model and then start to evaluate covariates, temperature, time, space, turbidity, or what have you. This is in the context of an endangered species or protected species, right? Like if you need to know where these things are, and then you can begin to answer other questions like how are they doing and how many are there. In the case of eradication or an invasive species, the key to any invasive is early detection so that you can either attempt to eradicate or at the least attempt containment. So early detection is the key. It's just like in fighting a disease, but trying to figure out how to control invasive species. You need to figure this out pretty quick. Next, so you keep going. I don't know where you guys are at in the animation. I'm looking at a green box. The next one is back to the original 3 questions here.
I have those.
Where are they? Okay, where are they? How many are there, and how are they doing? So the eDNA, again, it goes back to question 1. Where are they? Once you get there, and you can hopefully develop an occupancy model, you can then, what we've used eDNA to do is rededicate resources or refocus energy where the target organisms actually are. So then we can start to do it in more, we can start to utilize more intensive investigative techniques like population genetics and try to figure out a population size, get a census count. How many are there? in the habitat. Then we move on to the next step, which is how are those individuals doing? Are they inbred or are they reproducing successfully with a lot of variability? Again, eDNA is focused on the orangish yellow corner there. It's where are they? Next slide, please. And for the data aspect of this, we have found currently that our ability to generate data, especially with metabarcoding protocol, our ability to generate data has outpaced our ability to make sense of some of this data. To confidently analyze and interpret and compare across labs, especially when it comes to metabarcoding. qPCR analysis is a little more straightforward, and it's a little easier to analyze, and if everybody has at least some minimum reporting standards as to how they're doing sampling in the environment and how they're doing qPCR, we can at least work our way through and compare data across labs and across habitat types and waterways and species types and that kind of thing. So we have found that the bioinformatics aspects of data interpretation and communication they're coming along. There's some fantastic ideas out there. All the tools aren't readily available for everybody to use and work and at least have a common platform. The DNA reference data, this is stuff that the Workgroup had discussed in its first meeting. A publicly available California-based reference database with DNA database, positive controls, multiple genes that have been sequenced from individuals from within our state, within the habitat that we are attempting to protect, or the species that we're attempting to help. Even invasives, or the ones we're trying to eradicate, that would be extremely helpful in getting people to compare their data. And publicly available qPCR assays that are like primer and probe sets that have been submitted so that other people can at least compare theirs. Like I design a lot of assays and then come to find out somebody else designed an assay, but I don't have any way to go reproduce these assays and at least compare them and use the one that works best. Typically, I call people like Karen Goldberg. We call Karen Goldberg. We look on her website, and she has a lot of assays listed, and we pick up the phone and call her and ask her how her stuff is working. That's very helpful. A database, a publicly available database where these types of things can occur with some minimum reporting standards. That would go a long way. Next slide, please. This will lend itself towards interpretation. How do we interpret the data? We need to. Everybody's going to face the same problems in data interpretation. Coming up with some minimum reporting guidelines, and then discussing how the data can be interpreted will be extremely helpful. That's already occurring. This will lend itself towards scaling this technology up, because we all know that this is large. It's a huge state with so many different habitat types. It would be great if we had 1,000 people with a peristaltic pump and some silicone tubes and some filters, but at some point, some of the methods will become more standardized in filtration types and volume, these things, but scaling will become an issue. Scaling is not an easy thing to do, especially in the delta. That's something we deal with every day. How do you sample the delta meaningfully using eDNA? That's a problem we face every day. Some of you might not face that. Next slide, please. I'm not sure. There should be a big arrow going down to scaling. We have run into issues with being able to process enough samples fast enough with equal ability. Of course, you can speed yourself up, but sometimes quality is degraded with quantity. So how do you maintain quality and increase quantity? Well, automation is typically the way you go. How that's going to happen, we have a robot, but we can't hook our filter into our robot. So we're working on that as well. Automating will help with some of the standardization and QA/QC steps. Next slide. That helps you get more efficient. So you can make more observations on a larger landscape, which is important, especially with as fast as our habitat types and our environment as a planet is changing rapidly. It's happening really quick. So we need to make more observations more often in more places. Next slide, please. This will help to create larger networks, I believe. We need to create larger networks of sample acquisition, processing, and data interpretation, and then subsequent dissemination of that data in a standardized fashion. Next slide, please, so that we can actually create data networks and start to make large-scale observations on an ecosystem scale, if possible. Maybe those ecosystems are small to start, but we can start to link these things together. Next slide, please. And we believe that Genidaqs, with cooperation, a healthy dose of caution, and a greater understanding of how eDNA works, and an adherence to an agreed upon set of minimum reporting requirements, I feel the potential for eDNA will be realized, and we will have our, I put in this NASA JPL photo. I always love it when they land something on Mars and they get all excited and they high five each other. So hopefully we get to that point, and not the other picture, which I couldn't find, is the one when they crashed the lander on Mars, when the European Union and JPL were working together, and the guy at JPL forgot to convert meters to feet or feet to meters, and they crashed the probe into the surface of Mars. They don't show that. Everybody's all bummed out, and none of them probably worked at JPL again. But so hopefully we can all work together to get to that, point where we're feeling like we just landed a probe on Mars. And that is it. I went through pretty fast, so I'm ready for questions. I know we're past our time frame.
We had a question on your slide 14. On the slides, what did the two rows mean?
There's a high volume and a lower volume. So the bottom one is a larger volume than the top row. So I'm guessing 14. Go back to 14. I'm guessing that's the.
Yeah, it's the drainage area and sampling. It's with Eric Waits and Roy Martin.
Yeah, that's the model for it. So there's two volumes. Top row is lower volume. Bottom row is a larger volume. And I can't off the top of my head remember the volume. Sorry, guys.
But thank you, Greg, for this presentation. It was a great overview.
You're welcome. Sorry I had to do it so fast, but it's a lot of information to go through quickly.
Yeah.